I now have the pleasure of introducing Bin Wu from Agios Pharmaceuticals. You all have heard a lot about the IDH1 mutation and how it has it's one of the most common targetable mutations for our cholangiocarcinoma patients, and Dr. Kalin, I think, did a great job of laying the groundwork to explain some of the science that has gone into understanding IDH mutations in solid tumors, and Agios has very beautifully taken that work from the bench and taken it into the clinic. And so Ben is going to be speaking on some of the preliminary clinical data out of the phase 1 that was presented at ASCO last year, as well as some of the correlative work that was very brilliantly written into that study, and maybe even touch on their phase 3 that is currently open. Please welcome Ben.
Hi, good afternoon. So thank you, CCF, and thank you, the organizer, and for the invitation and introduction. And the agenda was arranged in such a genius way that this morning, Vikram gave us a fantastic and timely talk about the histology, and now Dr. Bill Killing has provided a lot of background in terms of science, so that sets the stage of make my talk a lot easier. So it's a pleasure to be here today and to present on behalf of Agios team the result from phase 1 dose escalation and expansion cohort in mutant IDH1 cholangiocarcinoma patient. So IDH1, one of the 3 isoforms of isocitrate dehydrogenase family of proteins, is located in the plasma. All 3 enzymes catalyzes the oxidative decarboxylation of isocitrate to produce alpha-ketoglutarate under normal physiological conditions. IDH1 and IDH2 mutations have been described in both hematologic and solid tumors. Specifically, IDH1 mutations occur in about 13 to 15% of overall cholangiocarcinoma patients. About a quarter of the intrahepatic cholangio patient carries the IDH1 mutation. Mutant IDH1 enzyme is catalytically inactive. However, it possesses a novel gain-of-function activities in this critical metabolic pathway. And the end result is that the accumulation of oncometabolite 2HG, and that activities in this critical metabolic pathway. And the end result is that the accumulation of oncometabolite 2HG, and that in turn leads to the dysregulated epigenetic rewiring and cell differentiation block. Ivosidenib, also called AG120, or in short, IVO, is an investigational first-in-class oral potent reversible targeted inhibitor of the mutant IDH1 enzyme. And this has been extensively studied in both hematologic malignancies in addition to solid tumor. And you probably know that at the end of 2017, Agios filed NDA for ivosidenib to be for the treatment of AML patient, a relapsed refractory AML patient with IDH1 mutation. So here we report the data from cholangio patient enrolled into the phase 1 dose escalation, the multi-center dose escalation and expansion study. The study began in 2014. A total of 24 patients was treated in the dose escalation cohort from doses starting 100 milligram BID to up to 1200 milligram QD. MTD was not identified. 500 milligram QD dosing was selected based on safety, tolerability, and PK/PD profile from dose escalation cohort. Subsequently, a total of 49 patients were treated in the expansion cohort with the 500 QD dose. So it's quite an effort to enroll 73 mutant IDH1 cholangiocarcinoma patients in about 2 years span. And considering that this is a rare disease and with the relatively low prevalence of IDH1 mutation. And the primary secondary objectives of the study are quite standard for a first-in-human phase 1 study with the focus of safety, tolerability, and MTD, with secondary endpoints of PK/PD, as well as the clinical activity. In the second half of my talk, I will also discuss the some of the translational work, specifically the morphological change and gene expression profiling of serial tumor biopsies collected from this study, and the correlation of the clinical outcome. So the clinical data has been presented at ASCO 2017 and will be again in a poster session discussed later this evening. And our Agios clinical colleagues are there and available to answer any of the questions you may have in greater details. In short, 73 patients, mutant IDH1 cholangiocarcinoma was treated in the entire study, and the ivosidenib demonstrated favorable toxicity profile with without dose-limiting toxicity. And the 2 patients discontinued study drug due to adverse events were deemed unrelated to the drug. So these are as of March 2017 data cutoff. So this is a summary of the PK/PD profile. Ivosidenib demonstrated good bioavailability, rapid absorption, and relatively long terminal half-life, which supports the daily dosing. And most patients reach steady state level in cycle 1, and the plasma trial level were maintained at above the predicted efficacious level throughout the study. And to the right is the 2-HG, the oncometabolite, which is actually a pharmacodynamic marker. And you can see that the 2-HG level were maximally inhibited rapidly in cycle 1 and maintained throughout the treatment and across different dose level, so that these data combined with safety and tolerability supports the 500 milligram QD dosing to be used for the expansion cohort of the study and the ongoing phase 3 ClarlDHy study. Just to remind that to you that most of the patients enrolled on the study had prior therapy, up to 2 lines of prior therapy. So primarily, this is a group of third-line mutant IDH1 cholangiocarcinoma patient. Therefore, it is really encouraging for us to that majority of the patients derive some degree of clinical benefit, as demonstrated by the overall 61% of disease control rate, including 4 PR patient and 41 stable disease patient. And this encouraging clinical activity is further depicted here in a swim lane plot showing the duration of the treatment. Each line represents a patient. The light blue line are the 4 PR patient, with the majority patient in green are the stable disease patient, and the purple line depicts the PD patient. And I wanted to draw your attention to the 6-month PFS rate of 38.5% and 12-month PFS 20.7%. This is really encouraging to us, demonstrating the durability of the drug in a heavily pretreated population. So these are the characteristics of the 4 PR patient. And as you can see that the PFS on the ivosidenib, 3 out of 4 patients has the PFS beyond 12 months, and that's far exceeding the their duration on their last therapy, which is merely anywhere between 1 to 3 months. So this really is a demonstration of the durable effect of ivosidenib as a single agent in this patient population. And this is just a study case of a 66-year-old female. To the left is the CAT scan, and you can see that the two large liver mass were reduced in size, as indicated by the red arrow, after 6 months of treatment. And this patient remained on treatment beyond 12 months. To the right is the demonstration of the 2-HG as pharmacodynamic biomarker was maximally inhibited in both tumor biopsy and plasma to the level below within the healthy volunteer range. So that demonstrates the target engagement of ivosidenib to mutant IDH1 enzyme. So I'm going to switch gear to the translational study. Thanks to the patients and investigators and Agios team who made a heroic effort in actually campaigning and collecting baseline and on-study tumor biopsies. We actually have a good collection of pre-treatment matched pre-treatment and then post-dose tumor biopsies. In total, we have 17 patient serial tumor biopsy assessed in histology in collaboration with the expert pathologist who gave a wonderful talk this morning. And we also performed the RNA-seq analysis of 26 patient pre- and post-treatment biopsies. So Dr. Deshpande showed an example of the histology this morning. That was a stable disease patient. Here's another example of the PR patient, even to an untrained pair of eyes. You can see the difference in terms of baseline. It's a very loose, heterogeneous histo architecture. Now, after 6 months treatment, you can see this very organized pattern, and in particular, this antler horn configuration, which indicate of increase in cholangiola pattern. And I also want to point out another feature is if you notice that at baseline, there's the pink area around the nucleus is fairly large, and that has actually shrank, which dramatically shrank after 6 months of treatment. So these are the two features that came out of the assessment. And the assessment of the 17 patient histology is now summarized in this table. So 5 patients have increased in histology, the cholangiola pattern. And interestingly, these 5 patients, as indicated by the blue line, has prolonged progression-free survival compared to those patients who has no increase in cholangiola pattern. So this is the first time we're able to link the histology change to a favorable clinical outcome. We do recognize this is a small set, the limited sample size, so additional patient sample and data are needed to increase our confidence in this intriguing observation. And so, as I mentioned, we also perform the gene expression profiling in an attempt to understand, along with the histology change, are there any molecular feature that would further give us insight about the change observed in the tumor tissue. This is a heat map. Basically, this is a log fold change of about 500 liver-specific genes for every on-study tumor biopsy over baseline. So to the right side is the red area indicates the upregulation of these 500 liver-specific genes in 3 of the patients who actually have increased cholangiola pattern. So as we know from the preclinical work with Shu from Dr. Bardisi's lab, that IDH1 mutation is blocking the hepatocyte differentiation. So now we have preliminary data from clinical tumor biopsy treated with ivosidenib showing the increase of liver-specific genes. So that really speaks to the potential mechanism of action of ivosidenib in reversing the differentiation block in mutant IDH1 tumors. And if you recall, we have 2 different features for the histology. One is the cholangiola pattern. The other one I mentioned about the visible plasma shrinkage. So with the gene expression profiling, this is another observation we had, is that the patients with cytoplasmic decrease show increased expression of some immune response related genes. So we are continuing and analyze the sets of data and to understand what are the underlying biological and clinical significance of these. So to summarize, ivosidenib was well tolerated and associated with favorable safety profile, and ivosidenib demonstrated encouraging early evidence of clinical activities in this pretreated population with IDH1 mutation. And we have shown that ivosidenib may induce morphologic and molecular changes in a subset of these patients using serial tumor biopsies. And we're continuing to advance our research, and needless to say, we are also actively looking into the genetic mutation, co-mutation profile. So we're doing each of the analysis separately. Hopefully we will have enough of data sets, so eventually we can do integrative analysis. So collectively, all of these support the further development of ivosidenib in mutant IDH1 cholangiocarcinoma. That brings us to the ongoing the global phase 3 study of ivosidenib in mutant IDH1 cholangiocarcinoma, and this is a Okay, so this is a randomized, placebo-controlled study in previously treated population in cholangiocarcinoma, and we are implement prospective selection of mutant IDH1 patient, and detailed information can be found on clinicaltrial.gov and also the Clarity study specific website. And again, our clinical colleague will be here later this evening to discuss in greater details. So, thank you.